Is this your first time studying anatomical systems? Or maybe you've taken anatomy and physiology, but you're just not sure what the MCAT expects when it comes to excretory system? We're going to explore exactly what about the excretory system is going to be crucial for your MCAT success. We'll begin our exploration with some background anatomy of the excretory system. Then we'll zoom in a little bit on the kidney, and then a little bit deeper into the nephron for its structure and function. And then we'll talk about the physiology of the countercurrent multiplier system and the role that ADH and aldosterone play in kidney function. In this lesson, we'll explore the roles of the excretory system in homeostasis as tested by the MCAT, which will allow you to answer questions like this one about the nephron. The main purpose of the excretory system is to filter the blood, so let's follow the circulatory pathway first. We start with the aorta, which is more than just the arch that's depicted on top of the heart. It actually carries blood through your trunk to all your main arteries. In this case, we're concerned with the renal artery, which carries blood to the kidney. The renal artery then breaks down into even smaller arterioles, which ultimately carry the blood directly to the kidneys, left and right. After being filtered, the blood returns through the renal vein into the inferior vena cava, which leads back to the heart. Now, anything that's filtered from the blood follows a different pathway. From the kidneys, it goes to the ureters, left and right. These lead to the urinary bladder for storage. And then finally, it's excreted from the body through the urethra. Now let's visualize the kidney anatomy more closely. There are two main layers to the kidney. The outermost is referred to as the cortex. The inner is referred to as the medulla. Now there's also a concavity on the side of the kidney referred to as the renal hilum. Now the renal hilum gives access to three main vessels. First, the renal artery brings in blood, then the renal vein carries it away, and lastly the ureter carries away urine. Now the ureter expands when it enters the kidney and forms the renal pelvis as well, which is the central cavity of the kidney. Now the kidney is made up of 1,000,000 small subunits, each carrying out the filtration process. These subunits are called nephrons. And you can see that a nephron extends from the cortex into the medulla. Now let's take a closer look at the structures around the nephron. Around the nephron, we have blood coming from the renal artery into these afferent arterioles, which are just tiny little arteries. Then they pass through the glomerulus, and the glomerulus is just a ball of capillaries. Then it leaves through the efferent arterioles. Eventually, it leads to the secondary capillary bed known as the vasa recta. Why are we focusing so much on the pathway of blood? Because in this case, the glomerulus is where filtration begins, and the vasa recta is where we see reabsorption of vital nutrients. Now that we've covered the vessels surrounding the nephron, let's take a closer look at the nephron itself. Just as before, let's start by tracking blood flow. We can see the glomerulus, or ball of capillaries we discussed before, is completely surrounded by the Bowman's capsule. Sometimes these together are referred to as the renal corpuscle. Why do you think it's shaped like that? Well, just like in digestive and respiratory systems, it's all about creating large amounts of surface area. As we move from the renal corpuscle, we move into the proximal convoluted tubule, which in this diagram is simplified as just a little straight part of the tube. But you may recall from previous diagrams that it's actually much more complicated looking than just a straight tube. Next, we move into the loop of Henle, which can be divided into two main portions, the descending limb and the ascending limb. Now the ascending limb can also be further subdivided into the thin and thick portions. We'll see why these are relevant later. Lastly, we go to the distal convoluted tubule. Remember, proximal and distal are just anatomical terms that give us a location. So proximal convoluted tubule is closer to the Bowman's capsule, the start of the nephron, and the distal convoluted tubule is farther away. And after all the processing has been done, whatever's left over passes through the collecting duct. And as you can see on this diagram, we're moving from an increasing ionic gradient as we go from the cortex, outer medulla, to the inner medulla. Now, why is it structured this way? Well, we have three main roles of the nephron. First, with the blood flowing in, we filter. So we have filtration into the Bowman's capsule. Next, we have secretion. Waste products the body wants to get rid of are then secreted into this filtrate. And then finally, all the vital nutrients and other resources the body needs are reabsorbed back out of the filtrate and into the blood. You can think of it just like cleaning out an old junk drawer. You empty out everything in the drawer first, then you piece by piece pull all the good things back into the drawer and throw everything else in the trash. The same principle applies to the nephron. Let's explore how and why filtration occurs in the renal corpuscle. You can see from this expanded diagram showing the Bowman's capsule and glomerulus that we have various opposing forces. We have the hydrostatic pressure of the glomerular capillary, which is opposed by the oncotic pressure, which, if you're not familiar with the term, is just a type of osmotic pressure based on protein concentration. It even uses the same variable, pi. Now, on the other end, we have a hydrostatic pressure inside the Bowman space, or Bowman's capsule, which is also balanced out by an oncotic pressure. Now, when we put all these together, we call this the Starling equation. So these are Starling's forces. In this case, the hydrostatic pressure of the glomerular capillaries is much greater than inside the Bowman space. So this component of fluid flow will be into the nephron. On the other hand, the oncotic pressure of the glomerular capillary is higher than the oncotic pressure of the Bowman space, so there's flow out of the nephron. But since the hydrostatic pressure caused by blood pressure in the capillary is so much greater, the net flow is from the blood into the nephron. So this is how we create the filtrate. We push fluids and solutes into the Bowman space, or the Bowman's capsule. The next function of the nephron we discussed was secretion. And remember, secretion is when we take things the body doesn't really want anymore and we put them into the nephron, into the filtrate, so that they can be excreted. So it goes directly from the blood into the filtrate. We can get rid of a lot of different ions this way, ammonia and urea. As you can see, these are all being actively pumped into the various portions of the nephron so that we can excrete them. Now, the amount and type of compounds that are secreted is determined by your specific needs of your body at the time. And so this is not necessarily going to be constant. For instance, if you have an increased protein intake, when you digest those proteins, you're going to get nitrogenous wastes. So you might see an increase in ammonia and urea after a protein-heavy meal. If your blood pH drops due to excess H+, that'll be secreted as well. One last form of secretion is when we have waste products that are too large to pass into the Bowman's capsule. In that case, we can also pump those directly into the nephron, into the filtrate. The final function of the nephron is reabsorption. That is to say, we're going in the reverse direction of secretion. Instead, we're taking things that the body wants to keep from the filtrate and putting it back into the blood, into your system. There are a few things that are almost always reabsorbed: amino acids, glucose, and vitamins. Then there are other things that are reabsorbed, like salt, depending on your body's current needs. Because of osmosis, salt and water typically go hand in hand. The mnemonic "water follows salt" can definitely be used to apply here. Also, ADH or aldosterone might be released by the endocrine system to alter your body's reuptake of water. Finally, urea is reabsorbed for maintaining the countercurrent multiplier system, which we'll see in just a moment.
This countercurrent multiplier system is how the body builds up a gradient used to separate types of solutes in the filtrate. We have two main portions of the nephron. We have these horizontal segments and vertical segments. The horizontal segments help us separate different types of solutes. The vertical segments help us focus on different concentrations and volumes of those different solutes in the filtrate. Now we're going to focus on those vertical sections. So first we have blood coming in an afferent arteriole and heading out in an efferent arteriole. But because of those hydrostatic pressures we talked about earlier, filtrate is pushed into the Bowman's capsule, and here you see the proximal convoluted tubule as well. Here we have salt and various nutrients being reabsorbed, as well as various things being secreted. But we've already discussed those earlier. Here we want to focus on what's happening in the loop of Henle. Here we've got water being pulled out. Then as we move up on the ascending limb, we've got salt coming out through ion channels in the thin part and pumps in the thick part. But it's all impermeable to water. So you can see that a concentration gradient, as it goes from kind of a cream color to yellow, it's showing us that there's an increased concentration gradient, or higher and higher concentrations as we go down the loop of Henle. Here again, we've got reabsorption and secretion, ultimately using the concentration gradient we've built through the countercurrent multiplier system with the loop of Henle to concentrate within the collecting duct our final urine product. Now you can see from this chart that we have a high concentration gradient. It would be even higher if you looked at, let's say, a camel or some desert-dwelling animal, because they really need to get all the water they can back out of the urine. For humans, though, we're going to go from about 300 milliosmolar, which is about the concentration of blood, up to 1200. So we've got 1200 milliosmolar, so a very high concentrated urine relative to our normal bodily fluids. The vasorecta is crucial in building this countercurrent multiplier system, named for the opposing currents you see here. On this animation, you can see exactly how it works. As the blood flows through the vasorecta, it absorbs this salt, this sodium that's being pulled out of the ascending loop of Henle. As it's pulled out, it increases the osmolarity of our blood in the vasorecta. Because we have such a high concentration and water follows salt, the salt reabsorbed in the ascending limb is now pulling out water from the descending limb. This gradient, although created by the loop of Henle, is eventually used by the collecting duct to concentrate the urine product. Earlier we mentioned ADH and aldosterone can affect the absorption of water in the kidneys, but we didn't exactly specify where. Aldosterone primarily works on the distal convoluted tubule, and you can see we have receptors for ADH mostly along the collecting ducts. Both aldosterone and ADH cause water reabsorption just through different means, so they're both going to give you a concentrated urine product. Now let's compare specifically how ADH and aldosterone are different. This is going to be important for the MCAT. They love to test the distinction between these two. First, we've got ADH, which is a peptide hormone, and aldosterone is a steroid. Triggered by high plasma osmolarity for ADH, we're going to absorb water. Aldosterone, triggered by low blood volume, this means we're going to absorb salt as well as water. ADH opens aquaporins in the collecting duct. Aldosterone increases sodium-potassium pump activity. So overall, the net effect for ADH is it reduces plasma osmolarity. It waters down your blood by absorbing only water. Aldosterone elevates your total blood volume by absorbing salt, and then the water follows salt. So the concentration doesn't really change in your blood for aldosterone, but it will for ADH. Let's review what we've explored with a practice question. Which of the following is responsible? Take a moment and answer. The proximal convoluted tubule. Most sodium reabsorption occurs in the proximal convoluted tubule, based on the percentages that we saw earlier. Let's try another. Go ahead and take a moment and answer this one about kidney structure. Longer loops of Henle, as we discussed with desert animals,
would give us a more concentrated urine product. Let's try one more conceptual question. Stick to the logical progression and you'll get to the right answer. Go ahead and try it now. Water building up in the extremities. This is also called swelling or edema. This can be caused by low oncotic pressure of the blood due to loss of protein. So what about excretion is most important for your MCAT? First, you need to understand the pathway that blood follows, as well as the pathway of excretion. You should also understand the basic anatomy of the kidney, the various layers, the vasculature, and the major central components, as well as the fundamental subunit that makes it function. Within that subunit, the nephron, you need to understand the various blood vessels there as well, the arterioles, glomerulus, as well as the vasa recta. You should familiarize yourself with the structure of the nephron as well as the pathway of filtrate as it moves through, starting with the glomerulus and Bowman's capsule, the proximal convoluted tubule, the loop of Henle, descending and ascending limb, as well as the thin and thick portions of the ascending limb, the distal convoluted tubule, and finally the collecting duct. Once you know the basic structure of the nephron, then focus on the functions. Filtration of the blood, secretion of wastes, and reabsorption of nutrients. Lastly, remember the countercurrent multiplier system and how it builds up a concentration gradient through the loop of Henle that allows us to selectively increase the concentration of various solutes within the waste product. Also recall that ADH and aldosterone both increase water reabsorption just through different methods. Thanks for joining me to flesh out the details of excretory system that are most important for the MCAT. Studying smarter using this resource as well as your other Kaplan materials can help you achieve your test day success. Good luck with your prep.